Pseudogymnoascus destructans is the fungus responsible for white nose syndrome, a disease that affects bats. Since its introduction to North America in 2006, it has decreased bat abundance by 10 times and has caused mass local extinctions continent-wide. This decline has had dire effects on both human health and economic output, stemming from increased pest populations and increased pesticide usage. Despite this severity, however, P. destructans remains poorly understood. Creating mutant strains with targeted gene deletions can help reveal the key factors related to virulence. However, the only established transformation method is slow and labor-intensive. Electroporation offers a much faster alternative. A 2019 study demonstrates this success in P. verrucosus, a close relative, and we adapted that protocol for use in P. destructans. Figure 1 summarizes our protocol. Cells were harvested, germinated, electroporated, allowed to recover without hygromycin, then transferred to selective media. Some were also plated on non-selective media to test viability. Figure 2 on the left shows the plasmid used, which contains Hyg-R for hygromycin resistance and AMA1 for independent replication. On the right is an example of what germinated conidia look like after 2 days in KSFM. Germinated cells have a visible tail structure. Figure 3 left displays successfully electroporated cells plated on PDA with hygromycin after 2 weeks. Looking to the right, this is a verification of hygromycin resistance. Untransformed cells on the top and transformed cells on the bottom were subcultured on PDA with and without hygromycin. Notice a lack of wild-type colonies on hygromycin. After confirming transformation, we tested voltage and DNA quantity as variables. Figure 4 shows viable cell counts across pulse strengths of 2.5 to 7.5 kilovolts per centimeter. Figure 5 on the left shows transformation efficiency across DNA inputs of 1 to 10 micrograms. The right panel shows efficiency across pulse strengths of 2.5 to 7.5 kilovolts per centimeter. It is important to note that colonies in figure 3D are smaller than in 3C, suggesting plasmid inefficiencies such as a weak promoter or a weak origin of replication. However, the plasmid is indeed functional, able to replicate independently as well as confer hygromycin resistance. We found transformation was most efficient at 4 kilovolts per centimeter and 1 microgram of DNA. Also, high voltages lead to cell death, which indicates that this is what causes the poor efficiency at high voltages. While more trials are needed to confirm this and explore additional variables, these results already show promise. Several genes that may be linked to pathogenesis have already been identified, and we aim to create targeted knockout lines soon.